and function. And to start with DNA structure and function, let me just open the slide where we were working on. So yesterday we were discussing about the double helix, how this works. So you know the structure of DNA. You have learned the model which was earlier given, and it's well known as a Watson-Crick model, where you have learned the complementarity of the structure, where one strand is complementary to the other strand. We have also discussed that there is a specific pairing of the bases in DNA, and that the logic why they are paired we have discussed. One thing is that you always have to accommodate within a particular diameter, and another you have learned about the hydrogen bonding. Then we have talked about that there is a semi-conservative transmission of the information from one generation to another generation. So when you look at the DNA replication, DNA replicates in such a way that one strand is always parental strand and next strand is the daughter strand. And that's why it was called as the semi-conservative model. Then to see such kind of mechanism of transferring information from one generation to another generation, the key factor which you have learned is that the DNA is the double helix, and whenever it makes another copy, it opens up and one of the strand becomes template strand, and then the next strand is being synthesized. So for this kind of activity, what you require is that opening of the strand. Here, I would like you to recall our second lecture where we have discussed about the non-covalent interaction in the biological system. Presume that if DNA helix are bound together by covalent bond, what will happen? It will not be possible to open up the strand and close it down, or to make the double helix to single strand and again make double stranded DNA. So what you have learned is that in the DNA helix, the strands they are held together by non-covalent interaction, and this non-covalent interaction is basically because of the hydrogen bonding, and that is given or intrinsic in the nucleotide sequence. Other than that, another kind of interaction which occurs within the DNA chains is by hydrophobic interaction, where nucleotides they are stacked together inside the core of the DNA helix. Now, coming to the point that if I have a DNA strand, if I give you a DNA strand in a test tube, can we make it to open up? Answer is yes. These double helix can be opened and again made in the double helix form by just heating and cooling them down. So here it has been very clearly demonstrated, and it can be done by experiment. You can also do it, that if I take DNA in a test tube, heat it, it will melt. And then I cool it down, then again it will anneal. Annealing means it will again form a double-stranded DNA. So if you look at this structure, what you find is that heating from 60 degree to somewhere 70 degree unfolds the DNA to say 50%. And then again, if you keep on heating, there will be time when the complete DNA strand can be separated out. So it means the DNA intrinsically, though present as a double helix, can be opened up. And once it is opened up, then it becomes a template strand for the synthesis of new DNA. So that part we'll discuss today. And the point you have learned here is that this can be monitored by spectrophotometer, and the type of, whenever the DNA they are stacked together, their nucleotides they are stacked together, they are having lesser absorbance. You can see the red one than when it opens up, which is the blue one. So single stranded will have a more absorbance than the double stranded, because the nucleotide, when they open up, when they are bases, they are open up, they absorb more ultraviolet light, and that's why you see the increase in the absorbance. And this phenomenon, the effect where the light is less absorbed because of the stacked bases is known as a hypochromism, and this is known for nucleic acid. So this is a graph very clearly demonstrate how you can perform it. Now question comes that in case of a cell, what happens? In cell also the similar phenomenon happens. Within the cell, they open up, but it does not require, heat energy. What happens, protein called as helicases, they use ATP and they disrupt the structure. So locally, the double-stranded nucleic acid strand opens up and then they replicate, duplicate, they transcribe, and all the phenomenon happens once they open up. But if you want to carry out the same reaction in the test tube, in the cell, in your lab, you have to provide heat energy or you have to provide ionic interaction. That's the way it can open up. Can you tell me, if I take a DNA strand and put them in a solution which has got, you know, uh, the where your pH is low, what will happen? Or let us not talk about pH, let us talk about the salt. Suppose I take a salt solution and put the DNA. In one case, in another case, I remove the salt solution completely from the DNA. What should happen and why? Any one of you? Normally, you know that in cell, we have certain amount of sodium chloride or the salt solution which is present in the cell. So in the cellular matrix, you will find, or inside the nucleic acid, near the nucleic acid, you will find the sodium ion present. Now, if I remove that sodium ion, what is expected? Can you hear me?
Yes, sir.
Is this question clear to you? In one case, for example, I have taken a scenario where I have put zero point 0.9 molar or zero point 0.5 molar sodium ion in which I have put the DNA. In another case, what I did is that the same DNA was taken, its sodium ion was removed by dialysis, and you have a DNA which is lacking the sodium ion. What is expected? The DNA strand, which we have talked about, let me just go back and talk about the strand. So here, can you see the double-stranded DNA? What I have talked about is that the sugar residue and phosphate residue, they are present on the outer surface, whereas the bases, they are present on the inner side. Now, what will happen because of the phosphate group, which is present on the outer side? What you expect is that it should have a lot of negative charges. And because of this negative charges, because of the phosphate group, these DNA and RNA, they are called as a nucleic acid. Acid term has been given. Now, what I'm doing is that we have a salt solution where the DNA is present. So this is the condition where naturally there is a certain amount of sodium ion. Normally, you will have sodium ion present in the cellular environment, and they are present on the surface of DNA. So what they will do is that they will neutralize most of the phosphate group charges, and that is why they are more stable. Now, if I remove this sodium ion from the DNA, what will happen? The DNA will have phosphate groups and they will repel each other. And because of that repulsion, DNA will not be stable DNA. So most of the time you find in the cell or whenever you have a DNA, isolated DNA, that has got sodium ion present on the surface of the DNA. And that's why they are more stable. So let us continue our discussion. So here you have learned that what kind of a DNA you have and how the TM can affect. So we have also discussed that the TM will depend on the concentration of your GC content and AT content. And as you know that the content of G is equal to C and A is equal to T, that is adenine is equal to thymine and guanine is equal to cytosine. So if I give you, say that if I say there is a test, you have a D and guanine. So how much will be cytosine? 40% will be cytosine. So what will be adenine? So 80% is your guanine and cytosine. What will be the your adenine and thymine? Should be 10% each, right? So these are a very simple thing you can calculate whenever given in a problem, and it's very simple to address. So if I give you one of the nucleotide, because of the nature of the complementarity, you can calculate the other type of the bases. And once you know the concentration of bases, you can predict whether the DNA is going to be more stable or less stable. And it means that if you have a lot of GC content, the DNA is going to be more stable. If you have a lot of AT content, that means DNA is unstable. And that, so it is the proportion of AT and GC content which determines the stability of the DNA. Now, there are some other structures which are also present in the DNA molecule. Today, in this topic, I'm not going to talk in details about the other part of the DNA, but the DNA structure which we have discussed is the most common DNA structure present in the cell. And that is why we have discussed them, and that is what has come up when the Watson Crick made the X-ray crystal and analyzed it. But this DNA can have other forms as well. So most common form of the DNA is the bDNA, which is found in the cellular environment. But you can also have part of the DNA which can have A form of DNA or Z form of DNA. They will vary in terms of their content, the organization, etc., which I'm not going to discuss. It's more of chemistry and more of the data. But other than that, let us look at the topography. Let us look at the type of the DNA. If you see the DNA, mostly we have talked about the linear DNA. Some of the DNA molecule could be circular. Have you seen circular DNA anywhere in
So
any
yeah,
bacteria?
In the bacteria.
Yeah, in the bacteria. So whenever you have a plasmid, you have the circular DNA. So there are many places where you can find circular DNA. And they are super coiled as well. Super coiled means their coiling is a very, it's like a spring. If I'm keeping a spring intact and I open up, they will release certain energy and they will work. So here we have also discussed that the superhelix of the DNA is helpful in certain functions, and the supercoiled DNA molecule is more compact in shape, so you can retain in a very small area. The other day we have talked about two things. Now try yourself and try to find out that whenever we were talking about the DNA structure, I'll just go back and give you some estimate, and that will give you a very interesting idea. Whenever we have talked about the DNA, you have, sorry, just to give an estimate, we have talked about the DNA. Uh, Here we have said and given you information that how many, so there are billions of nucleotides which are present in a DNA structure. That is what we have talked about in human DNA. We have also talked about that between the two nucleotide, the distance is 3.4 angstrom. So if I take a billion nucleotide and 3.54 angstrom distance between them, and you try calculating how much distance it will require, you will find there is enormous distance. And that is why it is most important that they should be packed thoroughly inside the, otherwise they will not fit in the your nucleus. So it's very important. So try calculating it. You will find the very interesting figures which will give you that why DNA should be a compact, should have a compact shape. And that's what can be attained when the DNA is a supercoiled. Other than that, supercoiling either hinders or favors the capacity of the DNA to unbind and thereby affect the interaction of DNA with protein, DNA with a protein, as well as DNA with another DNA molecule. Single-stranded nucleic acid often fold back. So when I talk about a single-stranded nucleic acid, I'm talking about the mostly RNA. And DNA, normally whenever there is another strand, they will form a complex structure. But RNA, they can adapt different shapes. So that is what we are going to talk about in the next slide. Here, we have talked about the different types of DNA structure. And so you have two complementary sequences where single strand come together to form a double helical structure. It is a stem loop type structure in RNA. In some cases, you may not have the Watson-Crick base pair. What is the Watson-Crick base pair? That means G is to C or A is to T. Sometimes there may be bulge which can appear, and there may be mismatch which may be appearing whenever the RNA is forming the structure. So look at the structure on the bottom right hand side. What you have, I have given you the details, whereas on the left hand side, I have given you a complex folded structure. So in general, when we talk about the RNA, you think that RNA is a single stranded molecule, and that is why it will not have any shape. But actually, RNA folds back on itself and creates a structure. And this structure is possible because of the kind of interaction they are having between different base pairs. So a single stranded RNA molecule may fold back, and once they fold back, they form a complex structure, and where the Watson-Crick pairing or non-standard pairing can occur. So there are different types of pairing occurs, and that's why the secondary structure for RNA can be stabilized. So this gives you the details and review of the previous lecture. Let us talk about the new one. So today we are going to talk about something which is about the DNA, how DNA forms another copy of the DNA, number one. And we will also talk about the, so let us go into details. So DNA can replicate, can form another strand. And you also know that DNA not only can replicate, they can also transcribe. So DNA can form another strand of the DNA. DNA can also form another strand of RNA. Though these things will be talked more in genetics and molecular biology, but right now I'll give you some introduction so that you have enough idea that how DNA replication or how DNA is going to make another copy of RNA. So if I look at a chemical reaction on the right hand side, let me just mark it. So here, how the DNA starts making another DNA molecule. So you have a DNA. Now, this is a, when I say DNA N, that means some of the nucleotides, they are pre-existing. And what is the DNTP? It is a deoxynucleotide triphosphates. When I say N, N could be any one of them, ATGC, any one of them. And when they are present, your N will become N plus 1 phosphate bond. They release the chemical structure of the DNA. So what you have, look at this structure, this part. This is the double-stranded DNA. So this acts as a template. So this is already part of the DNA. Now, this is the structure. This structure is unfolded structure. So you have just one strand. So what happens that whenever the replication occurs, uh, what will, I have given 20 proteins. That means there are different enzymes which are involved in the synthesis of the DNA. And most important enzyme which carries out the reaction is the DNA polymerase. So what DNA polymerase does is that DNA polymerase works here, and they will catalyze a reaction where DNA synthesis can occur. Giving the chemical synthesis, how this happens. So DNA chain is assembled directly on pre-existing DNA template. So this is the pre-existing DNA template. So first part on the left-hand side is the pre-existing DNA. Now what is happening? You have DNA polymerase in the cell, and you have deoxyadenosine triphosphate. And whenever it comes, what will happen? One residue, one adenosine is added over here. And that is the way the reaction goes on. Now, next time, what will happen? One deoxy GTP comes, argonisine triphosphate comes, and one G residue is added up here. So, this way, what you find is that the DNA chain keeps on growing. So, first stage, you added adenine. In the second stage, you have added guanine. And what is happening? It is having on a template or DNA which has unfolded in the part of the DNA. So part of the DNA is a folded one. The part of the DNA is a double helix. Part of them they reopen, and after opening this happens. So let us see how it happens. So Okay, so let us talk about this kind of arrangement which is present in the DNA. This is the same thing which has been explained. So this is the DNA template. This is the DNA template. Now to explain it better for your visualization, I have DNA strand. Now in this DNA strand, first thing will happen is that helicase will come and the DNA, part of the DNA will open. And suppose here, the some part is opened up. So what will happen here, the nucleotides, they will come. Now, once these nucleotides come, they are going to add up in the primer, which is unfolded. So this is the DNA template where you have a basis. Now this base is already present. Now what is happening? So here, look at this. Uh, so DNA template is there. The bases, they will align. How the bases will come? The base will come, which is a complementary base. So this is a complementary base which is already present. So this is a part of the DNA, pre-existing DNA. Now what is coming? Now one nucleotide triphosphate is coming. Sorry, yeah. So nucleotide triphosphate has come, and what has happened? This phosphate group is being attacked by the hydroxyl group from the primer strand. And what will happen? This rearrangement occurs, and because of the rearrangement, this phosphate PPI, pyrophosphate, is being released, and because of that release, this phosphate bond is formed. So what we call this phosphate bond? So between the two nucleotides, or between the two bases, what kind of a phosphate residue is kind of a the name? Any one of you?
Phosphodiester bond.
Yes. So this is the phosphodiester bond. So this is the one which leads to the synthesis of DNA. So what you have? You have a primer strand, the strand which was already present, and this is the base which has come. Now, once this bond is formed, this base is complementary to this, and the DNA chain will grow. So this is the way the DNA will be synthesized. So what you need, let me just go back and try to give you the reaction. In the reaction, what you require? You require the primer strand. Sorry, you require primer. So this is your primer. This is the deoxynucleotide triphosphate, and you require what else? DNA polymerase, because DNA synthesis is happening, so you will have enzyme which is the DNA polymerase. You require certain ions like magnesium ion, and you need certain temperature. So at that temperature, this DNA has opened up. After opening up, your strand has got exposed, where the DTNP can interact, and then reaction go ahead. Now I'm putting a question, and that question is related with your understanding of this. Suppose I have a strand, and this is the one which is present in the reaction mixture. So let me just sorry, let me go back to this. Look at the way reaction is happening. So this is the thing which is present in your solution. So you have in a solution, number one, number two, this is the another component. You will have certain ions, magnesium ion, etc. You have DNA polymerase. Now the DNA synthesis is happening. What I did is that what I can do is I change the base. Here, what I did is that uh, if I look at the base, in the base, you have here the if I say it is a deoxyribose, isn't it? You in this place you will have HH, right? What I did that in the chain king, so I take a base which is a dideoxy. So what I'm doing is that this is the 2' position, this 3' position. So I have taken a base which is 2', 3' dideoxy. Can you think of what is the dideoxy? So dideoxy means at both the position I don't have oxygen present. So here oxygen is absent, here oxygen is present. Now tell me what will happen. Any one of you? What will happen if I take a dideoxy in this reaction mixture? Think of it.
So it will terminate the strand.
Yes, it should terminate this strand. Why? Because suppose here in this position up to here the synthesis occurred. Now here instead of OH we will have H, right? Now if I have H, what will happen? Beyond this the reaction will not happen because this part is missing. And if this part is missing, there is no attack on this phosphate group which is coming from another nucleotide. And if this is absent, if OH group is absent, no further reaction will proceed. So this is one of the way by which, by terminating the reaction, people they started looking of DNA. I'm not going to talk about the sequencing today, but it gives you an idea that if I want to sequence a DNA and I want to know what are the bases which are present, so I know this base. As soon as I make another one which is which is having dideoxy, the chain will terminate. So by terminating the chain, we can find out which nucleotide is there. So if I have a 10 such residues, we can find out, and if I terminate at different time, you will find out what are the bases present. So you will learn this when you learn about the sequencing of DNA. Now let us move on further. Now question comes, is the DNA only which is genetic material? Answer is no. And now with the coronavirus, you know that RNA, which is the genetic material, and that's why the viruses, they evolved first before prokaryotes, eukaryotes, and that is where they were having the RNA as a genetic material. Not all viruses have DNA as a RNA as a genetic material. Some of them they do have DNA as genetic material. So this has evolved, and let us talk about how the RNA acts as a genetic material. If RNA is there, can they duplicate? Answer is no. So what happens that most of the RNA which is present in the viruses, which is the genetic material, but what happens that viruses, they do have a genetic element which encodes for which is enclosed in a protein code. So if I talk about a virus, what will happen is that virus will have, it will not have a machinery. So it is, suppose this is a virus, it is outside, it is a protein, and inside you have a genetic material, which is RNA material. It does not have its own machinery to synthesize, have a machinery, does the animal cell, it could be bacterial cell. So what they do is that they attack the cell and then they start. So there is a, they are not capable of independent growth. Now, one well-studied example of RNA virus is TMB virus. You will be learning more about this later on, which infects the tobacco plants and the leaves of the tobacco plants. So here, important class of RNA viruses could be retroviruses, where genetic information flows from RNA to DNA rather than DNA to RNA. This kind of information is also flowing in HIV viruses, AIDS viruses. Now you know about the coronaviruses. So what is happening here? So my question is that how we explain how the genetic material is formed and how the further growth of the or multiplication of viruses occurs. You know how fast viruses grow. But what happens? So you have a viral RNA. So RNA I am showing as a single strand. Of course, it can have a folding, etc. But this strand, they are in presence of reverse transcriptase. So what happens in case of RNA synthesis? There is a transcriptase. I'll talk about this transcriptase. Transcriptase is the enzyme which converts DNA into RNA. We will talk about that in next few slides. But in case of RNA, what will happen? The reversal will occur. So what will transcriptase do? Transcriptase will convert DNA into RNA. transcriptase, what it does, it converts viral RNA into DNA RNA hybrid. So initially what you get is that from the RNA strand, you get formed a complex of DNA and RNA strands. So what will happen here? The the nucleotides will come and they will make a DNA strand out of a RNA strand. Transcriptase does. And then it will synthesize another strand of the DNA, and that's the way you get double helical viral DNA. And then they will duplicate, they will form another strand of the DNA, and then they will do all the function. So looking at the information, how these information transfer from DNA, we need to understand. Actually, we have a little bit discussion on this, but there are steps which is involved. The information is stored in the DNA. What it does is that it forms RNA, and after forming RNA, so in the first stage, they will form RNA, and then a copy of this mRNA is formed, and then the information in the messenger RNA, whatever the message is there, they are translated into functional protein. So suppose I have this messenger RNA. Now this messenger RNA will have certain nucleotides. So there is a triplet of nucleotides. We call them as a codes. So you have these codons which are present in mRNA. What this codon will do is that they will recognize the transfer RNA. So these codons will, based on the anticodon present in the tRNA, so this will have anticodon which will be complementary to this, they will recognize. And each one will have amino acid. So this tRNA is having amino acid. So actually it explains the function. So a messenger RNA is a template for protein synthesis. So where protein is going to be synthesized? In the messenger RNA. And messenger RNA will be present in the, it will go assemble on the ribosome. And this is the codon. Here anticodon of the tRNA will come, bind over here. And so what will happen? You will have several amino acids, they will be present over here. So if I have several tRNA, they will come, each one will have a specific amino acid, they will be present here. Now once amino acid come in a sequence, they will have a bond formation. Energy will be involved for the bond formation, and then this sequence will be released from the ribosome, and they are the one which are proteins. So you have several, you have got a chain where amino acids are. So how this amino acid is formed? The information is coming from mRNA. And from where this information has come in mRNA is coming from DNA. So DNA transfer information to RNA, RNA transfer information to amino acid, and amino acid form protein, and that is what function. So that's what we are going to quickly discuss. You will learn complete protein synthesis separately. This is not the details of protein synthesis. This is just giving you how the RNA function. So an RNA molecule may be produced for each gene. So do you understand what is a gene? So far we have not talked about. Genes are, suppose I have a DNA, is a DNA molecule, and out of this DNA, this part of the DNA, if it functions for the synthesis of a protein, they will be called as a gene. So DNA is a general term. Out of this DNA, whatever the part which is coding for a particular protein, they may be referred as a gene. So gene and they are not different. Specifically present in the DNA. This mRNA is a heterogeneous class. Anyway, that is okay. Now coming to the tRNA part, they carry the amino acid as I explained here, and what will happen? They will lead to the peptide bond formation, and this peptide bond formation will lead to the synthesis of protein. So actually, these amino acids, they will come close to each other, and the peptide bond formation occurs. I have not explained and not going into details of this protein synthesis. That separate class. It will take 3-4 classes to explain protein synthesis. So what it means is that tRNA are variable, and that's why it says that they are different classes. So you have a tRNA which at least for each amino acid you will have a different tRNA, and they will be having anticodon as well. tRNA consists of 75 nucleotides. It's a very, very small RNA. So smallest part of the RNA is the transfer RNA or tRNA. Then you have ribosomal RNA. Where this ribosomal RNA is present in the ribosomes. And they are different, based on sedimentation. They have been given different numbers. And one molecule of each of this RNA is present in each ribosome. Now, coming to the cellular RNA and how it is being synthesized, you have learned how DNA is synthesized. The same way RNA will be synthesized, but what will be the difference? The difference will be that here the template is going to be different. And based on that template, your synthesis will occur. So you have RNA as a template, and then you have a ribonucleotide triphosphate. What is present in case of DNA? In DNA, you have deoxyribonucleotide triphosphate. Here you have a ribonucleotide triphosphate, and that's why they will be different. And what else will be different? Can you think of it? What will be difference in the ribonucleotide? Instead of thymine, you will be having uracil. So instead of TTP, you will have UTP. Okay. Now looking at the synthesis of RNA from a DNA template, what is this phenomenon called as? This phenomenon is called as transcription. When DNA synthesizes another DNA molecule, we call it as a duplication or replication. When DNA synthesizes RNA, we call it as a transcription. And the enzyme which is present here will not be DNA polymerase, but it will be RNA polymerase. So another difference came during RNA synthesis is that you will have RNA as a template, you will have ribonucleotide triphosphate, you will have UTP, you will have RNA polymerase, and then RNA synthesis will go ahead. So let us see what are the components of the RNA polymerase. They are, so when the preferred template in double-stranded DNA, the single-stranded DNA also can serve as a template. RNA, whether single or double-stranded, is not the effective template, nor the RNA DNA hybrids. I'll explain this a better way. Activated precursor or 4-ribonucleotide. Here you have same as in case of DNA, except for UTP in place of TTP. And divalent cation, as in case of DNA, is required here. So what it means is that suppose I have a DNA molecule. So the question here is that DNA, on one hand, synthesize another strand of the DNA, or DNA can also synthesize RNA. So DNA is acting as a template, and what you have is that it will synthesize RNA where some of the RNA molecules are already attached. So suppose there is a small part of RNA, and then it is going to synthesize the rest part of the RNA. So that is why it says the preferred template is a double-stranded DNA, and where it acts as a template, and then RNA, whether single or double-stranded, is not effective template nor RNA DNA hybrid. So basically, RNA polymerase requires double-stranded DNA, which will open up, and then synthesis will start. So RNA acts as a template, and another strand which is synthesizes the RNA molecule. So there is very much similarity in these two reactions. And if you look at this reaction, again, what is happening? Same way as we have discussed in case of a DNA synthesis, this is a DNA strand, DNA template strand, and what is happening that this is the base which is present in the primary strand, and what is being synthesized is that you have another ribonucleotide, and the thing you are looking at here is that it will have OH group in both of them. So what is the difference between DNA and RNA? While DNA, the nucleotide will have H over here, deoxy 1, where in case of RNA, this is having hydroxyl group at the second position. And then the chain will grow. So this is the primary strand. On the primary strand, this bond, which is a phosphodiester bond, is being synthesized, and that's the way your RNA synthesis occurs. Now, looking at gross level, how everything happens, the RNA polymerase, like DNA polymerase, takes instruction from DNA template. So suppose I have this as a DNA template. This shows a very interesting thing. So look at these strands. I have written one of them 5 prime to 3 prime, another one is 3 prime to 5 prime. Conventional way of writing, if I have to write a single stranded DNA, I have to write a strand which is 5 prime to 3 prime. Now I'm giving a question, and this is something which is asked sometime in a gate or other examination. Suppose I give you a strand which is this one. This is the strand. I have given you strand. I want you to write the complementary strand of this DNA. So this is the strand which is a coding strand, and it is forming another strand. So what will be there? The 5 prime start from here. The complementary part is in the blue form. So G will code for C, C will for G, G will have complementary C, G will have C. Same way, T will have A. So this way, one strand of DNA can form another strand of the DNA. Now, if I have to give in the writing just the strand which has been synthesized, now suppose I am giving the blue form. In conventional writing, I should not write like this unless I give 3 prime and 5 prime ending. Otherwise, how you will write the complementary strand of the black one? It should be written as starting with A, T, G, C, C, etc. Okay? Have you understand this point? To you? If I take this strand from where the DNA synthesis occurs. So what will happen? Of course, this blue one will be synthesized, but the strand will be in a different direction. So if I'm writing from, if it is a 5 prime, so the convention is to write from 5 prime to 3. So suppose I have to write a complementary sequence of this black one. The one which is synthesized should be written in a different way. So how I will write? I will write blue one separately starting with 5 prime. So G will have what?
Excuse me, sir.
Yeah.
Sir, could you please share your screen again?
Okay. If you're not able to see, I'll share it. Just a second. Uh, it is sharing. I'm stopping it sharing and again sharing it. Now, can you see my screen?
Yes, sir.
Okay.
Yes, sir.
Okay. So what I'm trying, so you should tell me whenever you are not able or then in the difficulty, please let me know. So this is a strand which I'm showing you. So till previous one you have been able to see? The previous slide, let me go back to previous slide. This was the previous slide where we are talking about the RNA synthesis. So here the same way synthesis occurs like in case of DNA. The only difference you have is that here the ribonucleotide will be there. That means OH group will be present at 2 prime as well. And the same way this phosphodiester bond is synthesized. Now coming to the next one, what I'm talking about is that if DNA has to synthesize another molecule of a DNA, so suppose this is the DNA strand. So what it will do is that suppose this is the DNA strand, which is the primer strand or the strand from which the DNA or replication has to occur. What will form? T will form A, T will form A. But what I'm trying to say here is that conventionally, if I write the strand which is given the blue color, it should be written from 5 prime to 3 prime, not from 3 prime to 5 prime. Is it clear? That means if I give you the black strand and I ask you what will be the complementary sequence, you should write it very differently. So first you write the way it is written, then you convert this into 5 prime to 3 prime. Now, what will be the 5 prime? It will start with AAA. Now, do you understand now what I mean to say? So these strand will be the starting point and the ending point will be this, and it will be presented in the form of 5 prime to 3 prime ending. Now, we are going to move on from here. This is a just convention which I was discussing. So now you have this coding strand and this is the template strand which has been synthesized. Now, whenever the RNA is synthesized, then again it will be 5 prime to 3 prime. So you see the reversal. Now what is happening here is you have adenine. Now if I have a DNA strand, what should have been synthesized? It should have been T over here. But because it is mRNA, you will have U. So if I look at the coding sequence in the template strand, so coding sequence and mRNA sequence is the same, except that you will have, instead of T, you will have uracil, right? Instead of thymine, you will have uracil. Otherwise, same sequence. But if I write it, because of the reversal, and that's the way it can be thought about. So this is what is about how RNA is being synthesized. So here, RNA polymerase, it takes instruction from DNA template. So DNA template is there. This DNA template gives you what kind of mRNA is synthesized, and this mRNA will give you what kind of a protein should be synthesized. So this way, we have discussed all the details about the nucleic acid. Let me know if you have any question or any confusion. Is it clear to all of you?
Yes, sir.
Okay. So in the next class, very quickly, I will, just I'm sharing my next slide, and this is what we will talk about. Today, I will not discuss. I'll just give you some introduction, and this is what we are going to discuss in the next class. So in next class, we are going to talk about lipids and cell membranes. And lipids, they are another class of biological molecules. These are another class of biological macromolecules. So you have several molecules, out of which we are discussing about the macromolecules. So you have learned about the carbohydrates, you have learned about the proteins, you have learned about the nucleic acid. Now we are moving on to lipids. And lipids, I'm merging with the cell membrane because there is a higher level of organization. So when I have talked about carbohydrate, when I have talked about proteins, when I have talked about nucleic acid, I was talking about a single type of macromolecule. But now what we are discussing is, now we are discussing a specific macromolecule, and then we'll talk about the cell membrane, which is a higher level of organization where you have a complex of lipids and proteins mostly, and sometimes there are a small amount of lipids which are associated with either lipids or proteins. So we will talk about the lipids and the cell membranes. And cell membranes, we will talk about what are the cell membranes, where they are present, how they look like. So you might have learned about the cell membrane, which is present in the bacteria, and you have different types of cell membranes. That means they may have outer membrane, they may have inner membrane, but there are a lot of similarity between all the membranes. Even if I talk about the cell, for example, if I take a cell, uh, have you learned about the cell structure in any course? Maybe in biological sciences, have you learned about the cell?
So, in microbiology.
Okay, you have learned about the bacterial cell. Okay, so in the bacterial cell, one thing extra you have is a cell wall. Animal cell, you will be having cell wall, which is the purpose is not to teach. Cell may not have so many developed, but as soon as you go to eukaryotes, you will have several membranes which are present. So one of the membrane could be nuclear membrane. So you have a nucleus. You may have a mitochondrial membrane. So you have a mitochondria where the membrane is present, which is the where the energy is being synthesized. You may have Golgi body where you can have membrane. So you have several places where the membranes are present. So what I mean to say here is that if I take the structure of any membrane, they have a lot of similarity. Suppose this is the membrane, which is a plasma membrane, which is the boundary of the cell. This is the second one which I am going to talk about is a nuclear membrane. So this is a nucleus membrane. This is a mitochondrial membrane. So there is a lot of similarity between all the membrane, and most of them, they are made up of lipids and proteins. So we'll talk about how these membranes, they are organized, what is their similarity, what could be the difference, and where and what kind of a function these membranes they provide to us. So with this, I'm going to sign off. If you have any question from the previous class or this class, you can raise the question. I would like you to be more interactive. Whenever you have any problem, anything which you are not understanding, please feel free to ask questions. So if you do not have any question, I would like to sign off.
Thank you, sir.
Okay, thank you. So one more question. Do you get the recording nowadays?
Yes, sir.
Yes, sir.
Okay. Okay, thank you. Bye.